With that, I'd like to go ahead and introduce our first speaker. We have Dr. Jessica Hartman from Cell Microsystems, and the talk title is Functional Cell Sorting, Automated Imaging, Sorting and Isolation of Single Cells, Clonal Colonies, and Organoids.
Thank you all. Christine, do you want to start my video?
Yes.
Perfect. All right, thank you all so much for having me here today and for the opportunity to tell you about Cell Microsystems and the CellRaft AIR technology. So I don't have to impress upon this audience that single cell biology is challenging, and really all biology in general is challenging. And we as scientists are only as good as or able to answer the questions that really drive us with the tools and technologies that are available to us. And they come in a variety of shapes and sizes, and they all have their various utilities. Some of those tools and technologies are cheap but laborious. Everybody can do limiting cell dilution, but it's not particularly effective or fast. Some technologies are high throughput but inaccurate or not amenable to getting the answer that you need. Some technologies start with live cells but kill them because the cells are fragile or sensitive or rare. Sometimes you can image the cells, like with high content imaging, but you can't actually collect the cells that are giving you the phenotype that you're interested in. On the converse, you can collect cells, but you can't image them. And more people are looking to do these types of complicated multi-parameter analyses. And some of these technologies enable those types of analyses, but they're often incomplete. So at Cell Microsystems, our goal is to make your life in the lab easier. And the way that we do that is by combining the best parts of a lot of these technologies into a single consumable and a single platform. So with traditional flow cytometry, you can very rapidly, with high throughput ability, isolate cells. You can have good cell viability, but you often need specialized facilities. It can be expensive, and these are time-consuming experiments. Similarly, with a droplet dispenser, you can isolate cells, you can sometimes image them, but you often can't get the imaging capabilities that you can with, say, a high-content imager. With a high-content imager, you can take great pictures, get great resolution, subcellular localization, but you can't actually isolate those cells. So with the CellRaft AIR technology I'm going to tell you about today, we give you the best of all worlds. We call it imaging plus isolation equals CellRaft AIR. I like to begin the talk with an illustration of how we can help you collapse your complex workflows using our tools. So if you've ever performed single cell cloning, you know that it starts with a population of heterogeneous cells that are transfected with your construct of interest, maybe overexpression or knockdown. You then select those stable cells, screen and select single cell clones, perform cell characterization, and expand and evaluate your resultant population. And this is often iterative, and you have to go back and forth a few times. In the traditional workflow, this means tissue culture plates, lots and lots of tissue culture plates, which turns into more tissue culture plates that you ultimately turn into single cells sitting independently in the reservoirs of 96-well plates. And then you characterize your cells using a variety of methodologies, high content imaging, sorting, next generation sequencing, and it's often very time consuming and laborious. In the AIR workflow, it starts with a single format consumable shown here. This is our CytoSort array. And that entire workflow can be performed on that single consumable on the deck of the AIR instrument. And that's what I'm going to talk to you about today. The core of our technology is the CellRaft. As you can see here in the bottom left, this is our proprietary consumable that we manufacture. It consists of a reservoir in the center, and if we zoom in, you can see that the reservoir contains these elastomeric microwells shown here. Each microwell contains a releasable magnetic polystyrene cell raft shown here at the top left. That means that the cells are growing on this cell raft in each independent microwell. However, the cells are sharing a contiguous volume of media in the reservoir, which enhances cell viability. In contrast to a single cell sitting in a 96-well plate all by its lonesome, which oftentimes can impact your phenotype and your viability, the cells think they are alone because they're in their individual microwell, but you get the benefit of the paracrine factors of the other cells in the reservoirs. The arrays are optically clear. You can enable imaging on these arrays with either fluorescent or by bright field microscopy. And then we can image, sort, and isolate single cells, colonies, organoids, what have you, by releasing those microwells. The consumable comes in several different formats. It's currently commercially available in the CytoSort single format, shown here on the left. That consists of a single microwell, a single reservoir, which contains anywhere between 10,000 and 40,000 microwells, depending on the size. It also comes in the quad format, which has 4 independent reservoirs, and each reservoir has between 1,600 and 4,000 microwells, depending on the format. The quads are particularly useful for multi-parameter assays where you have, for example, multiple drug concentrations or different guide pool RNAs, or you want to have your vehicle control on the same plate as your experimentals. We're going to soon be releasing the hexaquad, which is a 24 reservoir plate that fits into a standard 24-well SES format. And each of the reservoirs in the hexaquad have between 1,600 and 4,000 individual microwells. Our next product will be the 3D CytoSort, which has deeper microwells and a larger footprint for the cells to grow in, and that is to enable organoid or embryoid body culture for more three-dimensional type cultures. So to show you how this whole thing works, this is a cartoon of the CytoSort array in cross-section. The array itself contains your cells and your media. They're growing on the polystyrene rafts at the bottom. It sits above a release needle on a microscope objective. And then it will release that raft of interest, and a magnetic collection wand can dip into the array, retrieve the released raft, and then move it to your downstream collection plate. I'm going to play a little movie to show you what happens here. So the array sits on the deck of the instrument. You identify your cell or colony of interest. The needle comes up, pops the raft out of the microwell. The wand dips down, picks it up, and moves it to a downstream collection plate. In this example, we're showing a 96-well tissue culture plate, which allows for expansion of the cells off of the raft onto the floor of the 96-well plate if you're doing cell line development or you want to continue to grow those cells. You can also pick directly into a 96-well PCR plate or PCR strip tubes for downstream endpoint assays like next-generation sequencing or other type of endpoint phenotypic assays. The beauty of this system is that because the microwells are elastomeric, you can perform this release and transfer as many times as you'd like. You don't damage the integrity of the array by that release and puncture activity. So this allows for temporal isolation over the course of days or weeks, or as many isolations as you would like to perform. This is all occurring on the deck of our instrument, the CellRaft AIR System, which you can see here on the right. It consists of a fully integrated bright field and 3-color fluorescent inverted microscope, which enables for red, green, and blue fluorescence. It allows for fully automated, user-free, hands-free release and transfer of your cells or colonies of interest. It occurs completely in the instrument without any interaction. The machine itself can be upfit with an optional stage top incubator. If your workflows necessitate time course imaging or you want to continue to house the array on the deck of the instrument, that's a possibility with the incubator version. And ultimately, what the workflow allows is us to image, sort, and isolate cells or colonies from the array in a little under an hour. You can see what that looks like on the deck of the instrument here. The array is positioned on the left and your collection plate is on the right. So using the CytoSort array consumable and the CellRaft AIR technology, we can perform these incredibly user-friendly, fully automated workflows in a very rapid, easy-to-use manner. The workflow begins with the CytoSort array, which you use and interact with just like you would any other standard tissue culture ware. You prep the array for cell seeding, and then you can either apply coatings or not, depending on your use case. So some cells need Matrigel or laminin, something like that, but you don't have to because the cells will attach to the polystyrene surface. You seed your cells in your tissue culture media as normal, and that preparation hands-on time takes about 10 minutes. You then put the seeded array onto the AIR system. You can perform an array scan and image the entire array in as little as 10 minutes. You can then visualize and screen that array as many times as you want to. If your workflow is done for the day, you can put the array back in the incubator, continue to culture the cells as normal. Or if you're ready to isolate, you can choose your desired cells, set your parameters in the software, perform gating, and proceed to CellRaft collection. As I said, the imaging is hands-free, about 10 minutes, and the hands-on time of your analysis and gating can take as little as 5 minutes. Once you're ready to isolate your rafts of interest, you can sort and isolate them using our Cytometric software. You can isolate your rafts of interest, transfer and collect them to a 96-well plate, and you can fill a 96-well plate in about 45 minutes. Downstream, the possibilities are essentially endless. You can expand those cells over time. The cells will grow off the raft onto the culture plate and then can be passaged as normal to whatever subsequent assays you're doing. Or you could put them into your downstream genomics or lytic assay. This gives us a dramatically increased cell viability and shortens our time to endpoint analysis because the cells are much happier without these serial dissociation steps. So all told, hands-on time is about 15 minutes, and you can perform the complete workflow from seeding to isolation in a little over an hour. The brains of the system that drives the whole shebang is our proprietary software. This comes in 2 different interactive modes, either user navigation mode or cytometric mode, which I'll discuss shortly. The screen capture here I'm showing on the left is our user navigation mode. The software is incredibly user-friendly and intuitive, and it's one of the best pieces of feedback that we get from our customers is that it's really easy to learn and use, and we can typically get a new lab up and running on it in as little as a couple hours. If you are interacting with it in this fashion, it essentially functions as a live bright field or fluorescent microscope. You can manually click around the array itself, look at different fields of view, look at individual cells in the microwells, and sort of interact with it in a very manual way. You can define your imaging parameters, red, green, blue, binning, gain, exposure, whatnot, for your particular settings. You can stain your cells, do antibody staining, whatever phenotype you're looking for, you can analyze here. You can view the images in real time, or you can capture a full array scan. That will take a full image of the entire array in its complete format, and it will store it in a central database. The amazing thing about the software and the CytoSort arrays is that you can continue to serially image the plate over time, and you will get the exact same microwell in the exact same place every single time. If you've ever done high content imaging with a 96-well plate or a 384-well plate, you know you can go back to that same well every time, but you don't necessarily find the same cell or the same cells every single time. That's not a problem with the AIR system. Because of the way that this is set up, you can get an image of the same microwell every single day, every single time, and then all of that data is stored, and you can temporally click through all of your images to monitor colony formation, phenotype, what have you. When you are ready to isolate the rafts of interest and your experiment is over, it's as simple as clicking on your raft and clicking in the collection plate shown here on the right. So all you have to do is click in here, and you tell the software which rafts you are ready to isolate. You can also zoom in, take a closer look at your cells in whatever configuration you have, whether it's staining or bright field, and then you can make real-time decisions about the behavior of your cells. The other software mode that we offer is what we call cytometric mode, and this is our off-the-air version of the software. Obviously, as flow cytometry folks, this is going to look really familiar to you. After you've performed a full array scan, you can set your parameters of interest on the X and the Y axis, and you get a dot plot that represents all of the rafts in the scan that meet those criteria. You can toggle these depending on what you've stained with or what you're looking for. For example, we're showing here green fluorescence and bright field confluency at the end of the experiment. You can toggle the time points, you can toggle the parameters, and you can set as many gates as you would like. We have the standard tools up here, quadrant, line, ellipse, what have you. And you can set your gates on a variety of different modalities: bright field or fluorescence, intensity, confluency, cell shape, cell size, change over time, whatever you're interested in. Once you've gated your population and promoted those rafts, the software will automatically map all of your gated cell rafts to your downstream collection plate for isolation and retrieval. The off-the-air version of the software has a couple of really distinct advantages that I think are pretty unique. This is all happening offline, so this has the advantage of not tying up the instrument for another user. Unlike a flow sorter or another type of machine where it requires constant vigilance and someone paying attention to it and monopolizing that machine, this is all happening off the deck of the instrument on your own computer at your desk. So after a 10-minute array scan, you put your cells back in the incubator, you go analyze your data, and another user can come in, interact with the instrument itself and then go do their own analyses. So this is really good for multi-user cores or labs where you have a lot of people using the same instrumentation, because you get much more bang for your buck in terms of time on the instrument. The other big advantage is that, again, all of this is happening with your cells still happily adhered to the array, sitting in an incubator at 37 degrees. They're not flowing through sheath fluid. They're not sitting idle while you try to set your gates in real time to maximize viability. This is all happening offline so that your cells are happy, healthy, and adherent, all while this is going on in the incubator. So it's particularly good for sensitive cells or cells that are not amenable to being run through a sorter. So I've talked to you a little bit about the technology and how it operates, and now I'd like to show you some of the application possibilities that are possible with the AIR system. I love to show this picture. I'm a big fan of Good Eats and Alton Brown, and he's famous for his distaste of unitaskers. And he always says the only unitasker allowed in his kitchen is a fire extinguisher. And if you've ever worked in a lab, you know the prevalence of unitaskers in a lab. We've all bought them, we've all used them, and they oftentimes are really useful for that one thing, and then they sit idle on the lab bench because no one's interacting with them. The AIR system is the ultimate multitasker. It does a little bit of everything, and it does it really well. So you can interact with it in a variety of different ways, as a microscope, as a sorter, as an analyzer, as a cell-based assay platform, and all of those things give it a lot more power than, you know, a library prep machine, for example. So I'm going to touch on all these applications in brief, but we can go into more detail if there are questions. So the first workflow I want to highlight is induced pluripotent stem cell reprogramming. This is a hot area in science right now, and it's not always a particularly easy or streamlined workflow. So we wanted to compare the traditional workflow in 6-well plates against a workflow performed on CytoSort arrays in the CellRaft AIR technology. This was done in collaboration with the University of Virginia Stem Cell Core. We took plated fibroblasts and transduced them with reprogramming factors. After a week, the cells were dissociated, and then they were either plated onto embryonic stem cell coated 6-well plates, or they were seeded directly onto recombinant human laminin coated cytosort arrays. On the manual version, we performed visual inspection and colony isolation over the course of 3 weeks. And in the AIR workflow, we performed automated imaging and colony isolation in roughly the same time frame. So to show you what this looks like in the traditional workflow, after 13 days, we did see the formation of iPSC reprogrammed colonies in the 6-well plates. You can see them here in bright field. And we did live TRA160 GFP staining to indicate the presence of reprogramming, which you can see here. The clones were manually picked and transferred to new collection plates, but as you can see by day 21, they don't look particularly happy, and this is pretty traditional for iPSCs. The efficiency is less than 1%, it's very manual, and it typically requires an experienced user to monitor colony formation and transfer. So again, not a particularly robust or gratifying workflow. In contrast, when the workflow was performed on the AIR system, it was much more streamlined and much more amenable to user interaction. So you can see here a scan of the array. This is one of the microwells in the array at day 10. By day 10, we already have confluent cells on the microwell, on the raft in the microwell. We performed that live TRA160 staining directly on the array itself and then imaged it on the deck of the AIR instrument, and you can see those beautiful GFP positive TRA160 cells. We had to pick these rafts earlier than the 21-day time frame recommended by the protocol because they were already confluent and ready to go. So you can see here the raft has been transferred to a new collection plate, and in as little as a week, we're already seeing happy, healthy cells growing off the raft onto the floor of the new collection plate. So we're already seeing advantages in terms of usability. All of this is occurring in one platform, no need for other microscopes or other consumables, and we can stain directly on the array and image it. But the other big advantage is in contrast to the traditional workflow where you are hoping that you seeded your cells at single cell resolution and you don't really know for sure if the cells migrated into each other. We actually have the ability to track that over time. So if we look back, when this array was scanned at day 0, this is the same microwell. You can see there's the single cell right up there in the top right corner. By day 4, we're already seeing that cell begin to proliferate. And by day 10, there's that confluent cell layer that we see here. So there's a couple of distinct advantages to running this type of complicated workflow on the AIR system. First, we can get faster colonies that are more viable much more quickly, but also you can validate that you have a clonal population at the outset, and that really eliminates some of the downstream labor in terms of genomic sequencing or other types of manipulations to ensure that you have a homogeneous population. So the next workflow that I want to talk to you about is a head-to-head comparison of the AIR workflow versus flow cytometry in the CRISPR space. We have a lot of customers using the AIR system for CRISPR to streamline their cell line development. In this experiment, we nucleoaffected a GFP-RFP positive cell line with guide RNAs to GFP and RFP, and we performed the traditional workflow, 6-well plate recovery for 48 hours, and then we trypsinized the cells and collected them for sorting. They were sorted as single cells by flow cytometry, plated in 96-well plates, and we saw an average time to colony formation of around 6 to 10 days with a viability of about 29%, which is pretty typical for this type of workflow. If we perform a modified version of this, whereas instead of sorting the nucleoaffected cells by FACS, we trypsinized the cells and seeded them directly onto the CytoSort array with only 1,000 cells. In this workflow, we saw time to colony formation in as little as 5 days, and the viability was significantly increased at around 66%. This is an improvement in both time and viability, but also in the number of cells, which is particularly important when you don't have a lot to start with, or you're working with rare cell variants, because we could use 1,000 cells versus 1 million. If we perform what we call the idealized workflow, which is the direct to array workflow, after nucleoaffection, the cells were seeded directly on the CytoSort array with 1,000 cells, and we saw a much faster time to colony formation in as little as 3 or 4 days with an increased viability of 83%. And our customers routinely report that whereas with a flow-based single cell sorting workflow, they need hundreds of colonies to get a handful, they can routinely get 60 or 70 from a single plate from us very easily. Another key feature of this is that in addition to getting more viable cells faster and for the isolation step, you can also visually confirm your phenotype of interest very quickly using the fluorescent imaging. This particular experiment was performed in one of the quad arrays, which, as I mentioned, is really good for multi-variable analyses. In this particular experiment, the top left panel is a control, so no guide RNAs, and the cells are RFP and GFP positive. And in the bottom right, there's the double knockout. The cells are in fact negative for RFP and GFP. The top right is RFP only, so the cell is GFP but not RFP. And in the bottom left, the cell is negative for GFP and positive for RFP. So using the AIR system and the imaging capabilities, you can confirm your phenotype over time very quickly and then only isolate those cells that you are interested in for your downstream process, as opposed to flowing millions and millions of cells through a droplet dispenser and only getting a handful that you might need. In addition, we also have the ability to monitor clonality in this workflow as well. So in a typical workflow, again, you're hoping that you got a single cell and there were no doublets, and that if you had a clone at the end, it was in fact from a single cell. But almost always that necessitates genomic sequencing to make sure that you got the edit or the insertion that you want. So in this instance, I'm looking at the CellRaft organizer from our software. You can see 2 different rafts with 2 different clones on them, and they both look like colonies. But if we track back to day 1, you can very quickly see that one of these is clonal and one of these is not. So at day 1, you can see the top colony started from 2 cells, so it's not clonal, whereas the bottom colony started from a single cell, so it is in fact clonal. So again, it really is an advantage to be able to phenotypically monitor your cells over time in the system, and it does give you higher quality cells much faster because you don't have to be as concerned about the downstream confirmation of that clonality. So to summarize this, I think it's pretty clear how the AIR system can both streamline and make your cell line development more efficient. In the traditional workflow, this typically takes between 8 to 10 weeks of fairly laborious effort with lots of consumables and a lot of user input. In general, it has to have sequencing in order to confirm clonality and make sure that you actually have the clone that you want. And more importantly, this takes multiple replating steps. It's a lot of user interaction and a lot of stress on the cells, getting serially trypsinized, replated over and over again. In contrast, on the AIR system, we can go from a population to a clone in as little as 2 weeks. We can use a single consumable. This whole thing is happening on one plastic dish. We can visually confirm both clonality and phenotype, and it's incredibly gentle on the cells. There's no replating steps, there's no trypsinizations necessary, there's no expansion steps. I equate it to basically putting an ice cube from your ice cube tray directly into your drink instead of melting the ice cube before it goes into your drink. Another application where we've seen a lot of growing interest and a lot of utility is in the 3D organoid space. Organoids are typically not amenable to flow-based workflows because you can't easily flow a three-dimensional organoid through a flow sorter or a droplet dispenser. So in collaboration with the Magnus Lab at UNC, we've seen a lot of beautiful data coming from them about how they use our system to isolate these organoids. Their workflow starts with mouse gastric stem cells that have been pre-sorted using flow cytometry. So the cells are stained with whatever cell surface markers they're interested in, and the cells are seeded directly onto the array. You can see over here on the far right, that cell is going to become this beautiful organoid over the course of 2 weeks, and they performed that temporal time-based serial imaging of that microwell over time. The organoids can be released from the raft, from the array, still attached to the raft. I love this picture of this beautiful 3D organoid sitting on the surface of the array. And then that can be transferred into whatever downstream analyses you're doing. Sometimes they can be dissociated and replated back on the array for an organoid forming assay or other assays for cell-based assay work. More recently, they have been isolating these organoids directly into lysis buffer for library preparation and downstream RNA-seq. And we were quite gratified to find that the workflow is very amenable to this. You can get incredibly high quality RNA with great read counts out of this workflow. And they've been using this information to characterize the genetic heterogeneity of the organoids that form in their system. So I think this is a really nice example of how the AIR system can fit at the juxtaposition of several different technologies and really enable all of them to be more high throughput and more efficient. Either whether you're starting from sorted cells at the beginning or you're moving into an endpoint assay at the end of the experiment, the AIR system can really enable all of it.
And just to let you know, we have just a few minutes left.
Perfect. So the last assay that I would like to describe to you, I think, is a really good example of how you can use the CytoSort array and the AIR system to perform these types of cell-based assays. This is a unique ability of the technology that you can't necessarily perform with a traditional flow cytometry or a droplet dispenser. So this was work performed at UNC in the Albritton and Armistead labs, and they wanted to develop T-cell mediated killing and proliferation assays. In the top example, they pre-sorted CD8 positive human T-cells out of patients, and then they cultured the cells with target cells on the array and assessed the formation of apoptotic bodies over time. And you can see that at the 0 hour, there's no apoptosis, and by 6 hours, that GFP signal is indicative of apoptosis starting. The converse of this was they began with CD4 positive T-cells for proliferation assays, which they co-cultured with stimulating cells of interest. The cells were pre-stained with cell tracker red, which you can see down here, and then they monitored the loss of RFP as an indication of proliferation over time. So I think these two examples are really clear of the strength of the system and how you can use it to your advantage. You can co-culture cells of interest in the array. You can perform very elegant on-array phenotypic assessments, be it apoptosis, proliferation, what have you. You can do this temporally, where you can monitor it over hours or days, depending on your endpoint. And then ultimately, the real key here is the ability to isolate the cells that are actually giving you that phenotype. So in contrast to some experiments where it's totally endpoint, the cells are fixed, and you can't get those cells back at the end of your experiment, or worse, it's lytic, you can actually perform your live phenotypic assay, get the cells that gave you that phenotype, and then move them into your downstream analysis, whether it's expansion of that particular population, or in this case, cloning to identify the TCR sequences using sequencing. And really, again, that's the benefit of the system. All of these seqs that are coming out are fully executable on our system. And I like to say that it's any endpoint you seek, whether it's RNA with single-cell RNA-seq or TCR-seq, DNA, RNA plus protein, CRISPR, what have you. We're basically giving you the cells that are high quality that you want that can go into whatever assay that you're looking for at the end of your experiment. So with that, I'm going to wrap up, and hopefully I've shown you how you can use the CellRaft technology and our CytoSort arrays and the AIR system to image, sort, and isolate using our tools. Our CytoSort arrays are compatible with a variety of cell types, primary cells, cell lines, human, mouse, stem cells, what have you. The AIR system fully automates imaging, sorting, and isolation of desired populations for downstream applications. And really, we enable an absolutely endless range of application workflows, anywhere from CRISPR and cell line development to stem cells, antibody discovery, organoids, drug screening, and more. And with that, I will conclude, and I would be happy to take any questions. And my contact information is here, as well as the contact info for our East Coast sales manager.
Perfect. Thank you so much, Jessica. We really appreciate it. We have one question, and we have time for one very quick question. So Svetlana Mazel had asked, she said, amazing technology. Could you please specify the additional steps to be used in case of the non-adherent cell cloning? And we just have about 30 seconds for that answer.
Yeah, absolutely. So you can definitely use it with non-adherent cells, but it does require a little bit more upfront work. So you can either coat the plates with something to adhere the cells to them. So for example, a cell tack or a PLL, for example, and then you can transfer them to your downstream application if you're trying to keep them alive. If you are going for an endpoint assay, we've had a lot of our customers overlay a gelatin coating or a matrigel coating to seal those cells in the well if they're non-adherent, and then they can be released and transferred into the downstream application.
Okay, perfect. Thank you so much, Jessica.
Thank
that
you.
was a really wonderful talk, and that's all the time that we have questions for today. So thank you again. That was great.